Hi, good morning. My name is Gabriela Sanchez. Today is Saturday, November 14th, 2020. I'm going to explain how I did my wet mount of the cheek and the dental plaque. I did my previous video talking about the microscope and the different parts and how it's used. But for the next experiment, or the other experiment that I did, is I had to do the wet mount samples. So what I've gathered, my materials, I've gathered the staining tray. I have everything on a paper towel. I'm not sure if you could see, I have all this stuff down. And I'm going to talk about them. So I have the slide, okay, a clean slide. This is what I used, or one similar to this rather. I have the cover glass, and I have a pipette and the sterile cotton tip applicator. So the first step that I did was, obviously I washed my hands, disinfected my work area, gathered all of my materials. I obtained the first sample from the cheek. So I grabbed one of the cotton swabs and I vigorously rubbed the inside of my cheek. I did that vigorously, and once I had the sample on the cotton tip swab, I placed it on the slide. And I put it in the center and kind of did it in circular motions to get all the cells on the slide, or to get as many cells on the slide. Once I did that, I put it in the staining tray, and I applied approximately one drop of distilled water on there. I then grabbed the cover glass and I covered that area at an angle. So the cover glass was at an angle. This was straight and this was at an angle, and I slowly just kind of let it drop on top of the slide so that it could cover the area. Now there was some excess water that kind of seeped through the sides. I just grabbed the paper towel, damped it, or I kind of pat it dry so that it could absorb all that excess water. Once I did that, I placed it on the stage, on the microscope stage, and I adjusted the magnification so I could locate it and take a picture and upload it for my assignment. The same procedure was done with the dental plaque, with the exception that rather than vigorously swabbing the inside of my cheek, I did it at the gum line where my teeth were. So I vigorously swabbed that same technique, put it on the slide, add a drop of distilled water, covered it with a cover glass, and pat dry whatever excess fluid there was before I looked at it under the microscope. So it was getting easier to locate the cells, since I had already viewed the other cells that we were instructed to. So the bacteria spirillum, the yeast homount, the bacteriococcus form, the bacteria bacillus form, penicillin with conidia, and the amoeba homount. So because I had a little bit of practice doing that, I was able to locate the cells a little bit faster when I did my my swabs, my wet mounts. So that concludes my second part of the video for week 3. Thanks.